Just arrived and you already make it happen. Okay, so we will continue to the second part of our session, and we will start with Shai Morin from Rehovot of the Hebrew University of Jerusalem.
Okay. Good.
אתה רוצה שאני אגיד לך באיזשהו שלב או שאתה בסדר?
יש לי בעיה רצינית של זמן, שנראה כאילו כמה אני גרוע.
אני מבטיחה אז להגיד לך כשתתקרב.
So, good morning everybody, and I apologize, I'm not talking about climate change, but I do talk about adaptation, so at least this passage is the barrier. And just because I know I will run into problems with time, I'll just put this slide at the beginning and said this is large part of the PhD thesis of Ella, and that conducted the work with the help of Ksenia and Norah, and it was founded by the Israeli Science Foundation. So Oz gave a very good introduction, but although it's not dichotomic as it used to be, we can say that some insects are more specialists and some insects are more generalist, and its feeding strategy has its own advantage. And a paper that was published about 10 years ago clearly found that most species take the specialist kind of approach. That doesn't mean they feed only on one species of plants, but their range is quite limited, and there are only few species that are really true generalists, meaning that they feed on a large number of host plants belonging to a very big range of botanical families. But still, it's an open question of how actually generalist species are doing it. How are they capable of moving from one plant to the other all the time and being capable of using all these plants for feeding and reproduction? And there's no good answer in the literature of how actually generalist species are doing it. Some ideas are that they might harbor multiple versions of alleles. Each one are very good in utilizing one host, and so we are looking at a group of specialists that together we call generalist species. There are some examples in the literature for this, but I don't think that's a real good way to explain. On the other extreme, we can say that they can do okay on many hosts because they just have very narrow range, but this range of alleles help them use plants to some degree very well. Again, I'm not sure that this could be a good explanation because we know that some generalists are doing extremely well on many hosts, so it doesn't seem to be that they're just doing something, a mean kind of behavior. And so the idea that I think is more reasonable is that they live in their suitable host environment. They're not identical in any way, but they're kind of protected from selection because they produce selectively equivalent phenotypes. So they're not exactly identical, but they're doing very well on many kinds of hosts. But this is when they are in suitable environments. What happens if we stress them? If we take them to an environment which is less friendly? For example, they have to switch to a marginal suitable host. And so this is an example of a marginal suitable host. I'll come to it in a few minutes. And in this project, we had basically two research questions. First, what will happen both at the performance, gene expression, and genome regulation levels if this population, this generalist population, have to switch accidentally to a marginal suitable host? And what will happen, the same three ways of looking at it, if they have to stay there for multiple generations before they can switch back to a suitable host? So our model, already we see this model on here before, is the whitefly Domitian tabaci. It's a phloem feeding insect, I'll not go into it. And it's an extreme generalist. It can feed on hundreds of plant species belonging to dozens of different botanical families. In order to do our research, we established about 6 years ago an experimental system that started from field populations collected mainly from tomato and watermelon plants. The field population was brought to the lab, and since then, 6 years ago, we are maintaining them on their original hosts in this way, just to say that we are mixing. So we have 3/10 of one host and 3/10 of the other host, and each generation we collect insects from all the tents and move a mixture of all the tents to every new tent. So we try as best as we can to maintain the original genetic power of the population, the heterogeneous genetic power of the population. About 10 years after this, we took from these populations and moved them to a suitable host, cotton, and to a marginal suitable host of Anero pepper. And since then, we are now in generation 90, something like that. These insects only see cotton or only see habanero pepper, but again we mix between the cages in order to avoid genetic drift and other problems. So we still try to keep as best as we can the genetic diversity. So I'll try to show today 3 chapters. First, the adaptation process, how the population changed its adaptation, getting adapted to the marginal suitable host over many generations. So this will be just performance. Then we will visit very quickly the transcriptomic level of what were the changes that could be associated with adaptation to habanero pepper. And the last chapter, which will be hopefully the biggest one to show today, if I'll have time, is about the regulatory mechanisms, basically how the genome of generalist insects operate, and I'll come back to this figure later to explain exactly what we see here. So if we talk on performance assays, so every time we would like to do an experiments, we're doing experiments over the years. We take insects from the 2 selected populations and subject them to 4 treatments: the original plant and the other plant, in this case the marginal suitable host, and vice versa. We take the population that is getting adapted to habanero pepper and switch them and let them monitor their performance on habanero pepper and also on cotton, on the suitable host. We do it in the block design, and we measure basically survival and developmental rate. And during the experiment, we asked 3 questions. did adaptation occur? Do we see major differences in the performance? Like the habanero pepper population is doing much better than the population that sees habanero pepper for the first time. That's the first question. The second question is adaptation complete? Do they perform on the suitable host, or let's say the other way, do they perform on the marginal suitable host as good as they perform on the suitable host? And the third question, do they pay price for the adaptation? By getting adapted to habanero pepper, do they lose their ability to switch back to cotton, the suitable host? So this is a summary. It's a snapshot. We have more experiments, but I'm presenting those. We see here that we actually started from an old population and worked on it for a few years, but then decided that this population is not strong enough with the possibility of genetic drift and diversity. So we started all over again with the experimental setting that I showed you, and this is running for about 6 years. We can see here first that adaptation did occur. You can see here this is survival. You can see here that the survival of the habanero pepper population is significantly raising compared to the population that is experiencing habanero pepper for the first time. And you can see that it's evolving. You can see that they are getting better with generation that are getting, when we get to advanced generations. What we can also see here that it took them time to perform as well on habanero pepper than they are as they're performing, as the cotton population is performing on cotton, but they did achieve this stage. And we can also see here that they didn't lose their ability to switch back to cotton, like they're performing as well on cotton as the cotton population. The habanero population switching back to cotton is performing as well as the cotton population. We also are doing developmental rate studies. Basically the picture is the same, beside this experiment that didn't work well because we lost control of the temperature, but in all other experiments we can see that the population that is switching to habanero pepper for the first time takes much more time period to develop than the other treatments. So if we try to summarize the performance assays, did adaptation occur? Yes. Is adaptation complete? Yes, but it takes time. It took them like 6 years to really do as well as they are doing on cotton. And are they paying price during habanero pepper adaptation? No, they didn't lose their ability to switch back to cotton, even that they haven't seen it for 6 years. Okay. Now to the transcriptomic. So the same experimental setting, and this time we allow them to develop for one generation. We collect males and females separately and send them send it for RNA-seq analysis. When we analyze the data, we're looking for few ways of gene changes in gene expression. We are looking for plastic responses, meaning that already the original population switching for the first time to habanero pepper is expressing the genes as the adapted habanero pepper population. So this is a plastic response. They just have it in their original tools. Then we have then we have this one, which is in the theory of evolution. It's called accommodation, but basically it just states that they take the original response and just improve it a little bit in order to get better adapted to habanero pepper. Then we have this kind of stress response that is just noise. So they respond, but it doesn't give them any benefit. So when you look at the adapted population, it's switched back. So these are genes that are stress response genes that they are not providing any benefit, so they switch them back. And then there is the evolved ones, the ones that we don't see in the original capability of the population, but we see in the population that got adapted to habanero pepper. So these are the four, and if I try to summarize very quickly the transcriptomic results, you can see that from the four scenarios, the evolved scenario is the star. So there are some plastic responses, okay, but the majority of responses were evolved, meaning that it's not something that the original population can do. It takes time to do it. And if we try to understand what's behind adaptation to habanero pepper, so there are like 140 genes in total, but if we look at the major groups, we kind of identified three gene groups. One, and they are here. So the first one is a structural constituent of the cuticle. Not exactly clear why it's kind of a mechanism that allows the insect to feed on habanero pepper successfully, but it could relate it in the literature, it could be related to some kind of cuticle barrier enhancement, something maybe with desiccation resistance, because they maybe drink less and eat less, etc. Or hardening and thickening of the stylets, meaning that they're kind of have more powerful mouthparts. The second is oxidation reduction processes. This is also a very general response, so it's hard to say exactly what's going on. But it is related to raw scavenging genes. So part of the mechanism that the plant are using to fight the herbivorous insects is to attack them with reactive oxygen species, and the insects in response are producing enzymes and proteins that can scavenge the radicals. The first is the most interesting, and it's a downregulated group of genes. It's catepsins. Catepsins are proteinases, part of the arsenal of insects, and they are downregulated probably because they are effectors, meaning that they are secreted into the plant during feeding, and the plant are using them in order to identify that the plant are attacked, and they activate their defense system. So by downregulating the catepsins, the insects are escaping from activating the plant defense system. So these are the main findings. There were, of course, other genes that it's harder to explain exactly how do they contribute to adaptation to the habanero pepper. So the chapter 3 was to try to go beyond the transcriptome, to try to see how actually those responses are regulated at the genomic level. How, if I try to be really shachtzan, arrogant, to try to understand how generalist genome work. Okay, it's a very big question. It takes us years to understand, but at least we wanted to start asking this question. So we were we focused on three possible mechanisms, but just before that, we focused on changes in gene expression. So there was a background hypothesis all the time that we're not looking for changes in the coding region or something like that, because first it could be epistatic, it could be make a lot of mess, and second, it takes time to tune a change in an enzyme or something like that with mutations in order to let it work, and the changes we see are quite too fast. So we hypothesize we're doing something with gene regulation, down-regulating, up-regulating, which is much easier to change in order to see if it works. And the first mechanism we studied was an epigenetic mechanism, and I don't have time to explain what epigenetics exactly is. I'll just say that it's a way to look on the genome beyond the sequence level, but the packaging level, or how different regions of the genome are accessible to transcription and translation. So the genome is packed in different ways. Some genes are really open and can be transcribed easily. Some genes are really packed and cannot be transcribed at all. And every organism is using this trick in order to try to manage its optimal expression pattern. So this is epigenetics, and we studied DNA methylation. The second is kind of selection. So in a suitable host, you can have many versions of the promoter of a gene, but if you are getting to a stressful environment, you are using only some versions of this promoter that can support higher expression or lower expression. And the third mechanism is even harder to explain. It's something with gene networks, meaning that sometimes a change in one gene can make changes in many other genes. So it's very hard to detect because you see the change in all the other genes, but you don't know the mechanism until you find this kind of hub, the one that small changes in it will make changes in all the other genes. So we tried to see if we can chase all these three mechanisms. For the methylation study, we did again the experiments and collected samples and sent them now both to RNA-seq and to DNA-seq, and the DNA-seq was to trace methylation events by bisulfite conversion. And this is like a map, so I'll show you our results on DNA methylation. Then I'll show you the results on the promoter polymorphism, the SNPs. And if I'll have time, I'll say something about the networks. We're not finished with the work on the networks anyway, so I don't have much to say. So if we start with the methylation, we found that the genome of Bemisia tabaci has 11% methylation, which in insects is considered relatively high. This is the range in insects. So in insect, it can be considered relatively high. I will not talk about this figure, but you can clearly see that there is a population effect here. The populations coming from cotton and the populations coming from pepper are a little bit different. Unfortunately, we didn't find significant differences in methylations between the four treatments. What else we see here? We see that most of the methylation is concentrated in the exons and the introns. And this is known from other insect paper as well. And even more frustrating is that we didn't find any correlation between gene expression changes and methylation. This is what this figure is trying to show, that if you ignore, so we did find some genes that were differentially methylated, but we decided quite from the beginning that we are looking for a big change in methylation, and if we get significance, but the change is very small, we don't think that it could be biologically significant. So what this figure actually claims that there's no correlation between changes in gene expression and methylation, which was frustrating. But we did find one very interesting thing. So this figure shows you, so we took all the 13,000 genes of Bemisia and we ranked them according to their methylation level from low methylation to high methylation. On the y-axis you see the gene expression level. So first you see that more methylation, more expression. You can clearly see that heavy methylated genes are more expressed than hypomethylated genes, which is also known, nobody understands why, but it's also known in insects. Yeah. Yeah, I knew about it. And so, but we divided it into the three groups, and I'll talk about the hypermethylation and the hypomethylated ones. What we found very interestingly, that in the hypermethylation side, we found constitutive housekeeping genes, while in the hypomethylated group, we found environmental response genes which are differentially expressed. So this is new. This is what was not written before in the literature, that in order to be environmental response genes, you need to be hypomethylated. Why? We don't have a clue, but this is something that characterizes hypomethylated genes. If I'll try to go very fast just to show you what we did on promoters, so we asked if what I explained before, that we looked for changes in allele frequency. So maybe in suitable environments, they can have a lot of versions of the promoter, each one a little bit supporting a little bit different level of expression. But if you push them to a stressful environment, they switch to using only few promoters that are in one way, or can upregulate more or downregulate more. And so these are the three. When we went back to our gene expression analysis, so we looked for cases like this, which are plastic. We looked for cases like this, and we looked for cases like this in the frequency of the alleles in the population. And we were very happy to see that we think that there's something here. Without going into too much details of what we see here, we found in almost 30% of the gene that there is a switch to a different major promoter allele when we look about plastic from the original population. So CC is different from CP and PP. And even in the evolved category, it's almost when in some of the analysis it even reaches 40% that there is a switch in the promoter. So they're starting to use only some versions of the promoters that they actually had in the original population, which is much more diverse. And this is interesting because again, it wasn't reported before in the literature, and it's not surprising that many of these genes are hypomethylated. Many of the genes that are hypomethylated. One quick thing on the networks, that it's very programmatic to work with gene co-expression networks, because all the programs that we are using are actually looking for connections, like correlations between gene expression. They are not causative. They don't identify a hub, the Dremelab hub, like a transcription factor or a heat shock protein or anything that is classical in the literature. So we are still struggling, but just to be a little bit optimistic, if we look at these two, which are really characteristic of adaptation to habanero pepper, we can see here again the genes that were previously treated, sustained the catepsins, And here we see the cuticle binding proteins. So we just need to figure out how we take the hub out and try to understand more if there's something with gene network involved as well. So just to summarize this part, nothing with methylation or epigenetics with the behavior of generalists. Could be that there is a promoter allele selection, and we don't know yet about what's going on with gene networks. Thank you very much.
Yeah, so thank you very much, Shai. I find it's fascinating. One quick question before we continue, or if you are you're staying, right? So we can catch you all afterwards. So Shai is here, and I'm sure that there are a lot of questions to answer. So we will. Oh, sorry. Be careful. So we will continue immediately to Tzlil Labin.
It doesn't want to shift the presentation more.
No idea what's happening here.
See how too many open stuff?
How many years do you need to teach in order to know that?
Hi everybody. So I'll be speaking about flower color, insects, the synchronicity between them, and how it blends into the wider topic of global warming. So they say timing is everything in life, and when we talk about flowers and pollinators, they choose when to be active or how to time their activity according to different cues. For instance, the day-night cycle, rainfall, temperature. And these cues help them time the different stages in life, both for flowering stages and pollinators. And when we imagine a plant and its pollinator, so we often see them synced in their timing of activity. But if one of these cues shifts or changes, so perhaps they don't exactly have the same sensitivity to these cues, and then we might see different reaction norms of the plant and its main pollinators. And then we have an uncoupling of phenology and a mismatch between the flower and its pollinators. So what happens when we look into color? So first of all, we're talking about the species level. And within the species level, sometimes we have species having diverse traits, and these traits can directly or indirectly affect the coupling with the pollinators, the synchronicity with the pollinators. And specifically, I'm going to be talking about color. So this is a diverse trait we're going to focus on. So why do flowers even use color? Flowers use color for various reasons. For instance, to signal pollinators, to attract pollinators, and since different pollinators have different visual capabilities, so they can use different colors. And colors can also be used for adaptation to environmental stressors. So for instance, some colors are more adapted to UV, drought, and so on. And we're going to be talking about flower species that has all these various colors. It's flower color polymorphic, and it's very beautiful. Anemone coronaria, kalanit mitsuya. So it's not just wide in its color range, but it's also pretty vast in its distribution range. So we can find kalaniot, anemones, from the north to the south. And we actually, in our observations in the field, we found that we have these color polymorphic fields occurring from 450 millimeters of rain and above, but we don't find them in the desert. So in the desert line and in the semi-arid areas, we only find the red morphs. So that's the first clue for adaptation for drought. But the second thing we found that the red morph also flowers later, and actually the red corms from the south, the red plants in the south, they flower latest. So when we look at the flowering season, early in the season, when it's closer to the winter, at the beginning of the winter, then we find that the non-red colors dominate the field, but as we progress closer to the summer, by the end of the springtime, then the color dominating the fields is red. So we conducted a controlled watering experiment, and we collected corms from 3 different colors from the north in the Mediterranean area, and we grew them under watering resembling their home conditions, high conditions as in the Mediterranean area, but also kept them watered under arid conditions or low watering conditions as in the southern sites. And we did the same for anemones, red anemones we collected from the south. We grew them under dry conditions, but also under conditions mimicking the Mediterranean area. So first of all, what we found for the red morph, it was that regardless for where it originated from, whether it was from the Mediterranean or from the south, the corm grew larger under arid watering treatment, under low watering treatment. And this is what we found. So the weight ratio on the y-axis, year 2 versus year 1. And then we see that, we can see that the brighter color is the low watering treatment, and the darker color is the high watering treatment. And they obviously grew more under the drier conditions. And we found the opposite for the non-red corms. So purple and white grew larger corms under the improved watering conditions. And then when we look at the phenology, so I'll walk you down this one because it's a bit complex. So on the y-axis we have the number of flowers, and on the other y-axis we have the water accumulated during the treatment. And the gray line signifies the watering that they received during the treatment. So we see that in the low watering treatment, mimicking the arid conditions, they got a bit over 300 millimeters of rain, and in the high watering treatment, it resembled over 500 millimeters of rain. And basically, the main point is that it didn't affect the red ones a lot. They still started flowering around January. They ended their flowering around mid-March. They extended their flowering a bit more under the improved conditions. And their peak flowering was around the beginning of February, which again didn't differ. The water treatment didn't differ. So, if you wish. And then we added in the other colors, and we see that first of all, the first thing that pops into the eye is that white and purple flower before the red colors, also under controlled conditions and not just in the field. And they do it regardless of the watering treatment. Under the improved watering conditions, so they extend their flowering season almost until April. But another interesting thing was that they advanced their peak flowering under the improved conditions, and they reached their peak flowering at the second half of January. And if under the low watering treatment, they actually peak along with the red morph, and that can enhance competition. So as for the timeline, the non-red flowers before the red, and we also found that in all colors, the larger corms flowered earlier and for a longer duration than the smaller corms. And since the corms of the red flowers from the south were smallest, so they flowered latest. They had the shortest duration of flowering. And then we're going to make it more complex and add in the insects. So when we looked into the proportion of visits to the flowers, so we saw that flies and bees make most of their visits to our flowers early in the season, and they reduce their visits to the flowers as the season progresses. And we have a mirroring effect for the beetles, which visit our flowers more as the season increases. And the interesting thing that we found that most of the visits early in the season were by flies and bees were to the non-red flowers, and late in the season were by glaphyrid beetles to the red flowers. So that means that in order to enhance the probability of pollination, if you are a non-red flower, you should coincide with your pollinators and flower early in the season. And if you're a red flower, you should try and opt to flower late in the season. So we said the non-red flowers are limited to the Mediterranean area. Corm growth, corm weight gain is better under high watering conditions for the non-red and low watering conditions for the red. And the timing, well, the smaller the corm, the later the timing of flowering. Red flowers later, southern corms are smallest and flower latest. And for pollination, if you want to have higher pollination success as a non-red, you should flower early, coinciding with your main pollinators, and late for the red. So, are we expecting a monochromatic red future? Under arid conditions, that may slow the corm growth of the non-red, and if the non-red will have smaller corms, it will delay its flowering, and then we expect it might lower the synchronicity with its main pollinators. But if there will be a shift or a delay in the flowering of the red ones, it'll just enhance its coupling with its main pollinators, and that would result with an advantage for the red colors. So color matters, and red morphs seem to be more resilient to drought. If a delay occurs, it's better for the red ones, but it's not as good for the non-red ones, which can harm their interaction with their main pollinators. Climate change may delay flowering, threaten polymorphism in the areas which are more expected to global warming changes, and disrupt the nutrient supply for those early pollinators that are based on anemones. And thank you for...
Yes, so thank you, Tzlil. We have plenty of time for questions.
I have a question. You say that the color is reflecting the insect that pollinate. Is there a difference in the pollinator or the types of pollinators that you find in the north and in the south? I mean, is there? It's the chicken and egg thing. Who is determining what? The flowers determining the pollinators, or the pollinators determining the flowers?
So,
or am I talking shit? I don't know. It's possible too.
We placed traps alongside. I didn't show it, but we placed traps alongside our flowers, and the composition stays fairly similar along the season, but the visitation to the flowers differ. And what I think, perhaps the bees over the season, we do have a larger amount of bees, but their proportion is smaller, and I think they just go visit the other competing flowers later in the season. But we do have all composition of flowers and a similar composition along the gradient. The activity times are different. So the beetles do emerge later, and they're linked to the temperature. So actually, early in the season, we hardly had any beetles in our captures, and late in the season, we had more beetles.
So if you wanted to check my assumption, you would have to have early beetles.
Yeah, maybe.
Or modified
Or paint the color, the flowers.
But in that, I didn't say, but that experiment that we did to check the visitors, so we actually took, we plucked flowers, and we suggested all three colors in the desert and in the Mediterranean area in the center and in the north. So we gave them the opportunity to choose. The interesting thing maybe was that actually in the south, early in the season, bees, yeah, it was early in the season, I think. No, all along the season. In the south, the bees preferred the red and not the non-red. So overall, they preferred non-red, and in the north, they specifically preferred purple. But in the south, they preferred red. And I think maybe it's due to their learning capabilities. Glaphyrid beetles were very strictly only going to red flowers everywhere across. So they're more specialized to that color.
Okay, yeah. Peter, you want to?
Thanks. You show the difference in the corm size between the colors and the dates. I wonder if there's a difference in longevity, how many of them are closer to annual life and perennial, well, not perennial potentially, but between the colors or between south and north for the red ones. You know, this will play with how they allocate resources to flowering or maybe to...
I don't know about longevity. I assume that a big part of the corm size is the fact that they have less nutrients and less rain in the south. And I think that if we would have done these experiments from a seed, it would have helped us to eliminate the factor of the corm size, because then they would grow from, you know, and not, because we took them from the field and basically we already had these inherent differences in the corm sizes. We didn't check longevity, but the another thing that is interesting, and I talked to you guys before about it, was the fact that in the literature they say that perhaps the populations in the south also use vegetative reproduction, and that may also play a trade-off there. So I don't know about longevity.
Okay, so thank you very much again.
Can I get a last question? Okay?
Yes, okay.
What about the volatiles? Talking about color as a trait, but there's also volatiles as a trait.
Yeah, we didn't check that. We checked many things, we didn't check that.
But it's going to happen now after this session because there is already
Okay. So it's the audience that's also to say that.
Yeah, with Norah and her experiment, she checked the anthocyanin levels and maybe how they affect.
It's the same.
Yeah, we didn't check volatiles. We checked the soil.
Very
we checked
simply,
the
it was spread artificial flowers.
Maybe we can do it together, and yeah, I don't know.
So, thank you very much again. So we are going further again and have the same problem. What's the secret? What was the secret? How do I open? Can you please? Okay, so Kliot from the Volcani Institute.
How long do I have? You have 50 minutes with the question.
15 minutes.
Yeah, do you want me to signal to you? Or?
Yes.
Okay. I'll wait.
So is this even on? Should I stay here?
Okay, stay here just to finish reading.
Talk? Does it work? Do I need to talk? Can you hear me?
Yeah, I think you, if you can look the...
Okay, hi. Adi, Volcani Institute. And I'm going to talk about, like it says, outbreaks of the two-spotted spider mites in Israel during the hot season. Well, that's more like, this is like what brought started the research, but it's the title, or I'm going to run with it. So I think I'm using this slide from someone else's research, but I think it really describes a lot of what we've heard here today. All these different phenomena that can happen over climate change between insects and plants. And I'm going to touch on a few of those today. First, increased number of generations of the pest here. I should say, Volcani Institute, we're dealing with agricultures, we're not going into nature. These are pests, these are the enemy. So we're going to talk about increased number of generations of pests are happening with climate change, expansion of their geographic range, desynchronization between the pest and its predator. And I'm going to add a little thing of my own that I'm going to touch on today. Changes in the pest's physiology. Of course, it's the increased number of generations and everything is because of those changes, but we're going to see other aspects of these changes in the physiology and how they're kind of like giving us trouble. So, as I said, I'm talking about the two-spotted spider mite. It's a mite, that's not an insect, alright? It's an arthropod. It's just a little bit, just to show you, yeah, it looks very different. It's from the same family as ticks, although that's under debate, human debate. I think mites don't care. And they are teeny tiny. Just to show you here, can I get the video to work? How did I do that? How did we get the?
I hover.
Yeah, oh, yeah. I just needed to get you. But you see this flower and you see all these teeny tiny dots. So the largest adult female will be something like 0.2 millimeters in length. So we're talking about very minute creatures, and they're called spider mites because they weave webs. There's nothing else to get to do with this. So in Hebrew we call them the web mites, Kliot kurim, which is I think a more accurate name. So you see how small they are, but look at the infestation levels that we can get to. So here we have, it's pretty rare, but when we do get them in artichokes, then they kill the plants. So you see these little red piles, these have thousands and even millions of mites going to them and trying to finish with this plant. I don't know if you can see very well in this lighting, but this plant is all like silver color. They ate all the chlorophyll, all the plants around it are all much greener. They're done with this one, they want to go away. So they form these groups and hoping for the wind or someone to pass by and move them on. So, but this is just to show you like, to horrify you with the hormone pests, you know. And here, if we focus on these, you see that each is made like a lot of insects. Well, no insects, a lot of arthropods. And in Israel, at the moment, they're mainly an issue in peppers and in tomatoes. Again, we see here the symptoms on the tomatoes, so they feed on the epidermal cells and eat all the chlorophyll, so we have these white blotches. Same in the tomato. On the what is this leaf? The right, you see again the silvering of the plant. Right? It loses so much chlorophyll and it loses a lot of water. Over the hot season, it's a big issue. Now, in general, about their life cycle, it's very short. Under the right conditions, which it means like 30 to 35 degrees, we can have an egg-to-egg cycle within 7 days. So I don't know if you know. So for instance, for a farmer, if he's coming up to harvest, he cannot spray anything when it's about 4 weeks before harvesting tomatoes, maybe I'm talking here. So in these 4 weeks, he can have 7 generations of these mites. While each adult will still be alive, an adult can survive for another 30 eggs, laying at least 30 days, laying at least 1 egg a day. And on the peak days, you can lay even 10 eggs a day. So yeah, we have issues here. I wrote poikilothermic, like all arthropods are, but in this case, we are talking about a very thermophilic even pest. It loves the heat, which I'll show you in a bit as well. And to make matters worse, Shai earlier was talking about generalist insects versus very specialized insects. Also talking about that. Here we have a very, very generalist pest. And I have to say, so this is a list formed by Iraq, not the country, the Insecticide Resistance Action Committee. And they said these in 2020 were the top 15 most dangerous pests we have in terms of pesticide resistance. And who is the star? To be honest, I'm not sure if you look just below it. The next one has more cases, but these are the number of reported cases of different chemicals that this insect or pest is resistant to. So by in 2019 we already had almost 520 different chemicals that this tiny little pest is resistant to, and we think it's because it's also a generalist. So it can live on so many different plant hosts, it already has in its arsenal all kinds of ways to deal with different chemicals. So, still introduction. We said we will talk about the geographic spread. So this is a study done in Europe, and a mite, not the one that we were talking about earlier, but advanced, which is a problem for them, not so much for us. I don't know, again, if you can see. In black is where they actually documented cases of this mite, and then with different colors is the expected spread of it as climate change around the Mediterranean basin is going to continue, the climate is going to continue to rise. And this is what we're expecting to see by, I think they were projecting to about now. So we're in there now. So, and if this is where we are, yeah, we have a lot of red. So what are we doing? what we're seeing is that we get more mites now. This is why they took on an ecologist. And we're seeing a lot more damage over summer. And we were thinking, why? Why are we seeing it? So first of all, it's because our natural enemies are failing. So this is a study by a group from Europe, not mine. But what we can see that in general, you see the longevity of the pest. We're measuring it over here over 10 days of an adult. No mortality, almost no mortality in the pest, but over there is a predator, a mite that feeds on another mite. And slowly we have reducing rates. And if you see it in the red, where temperatures are higher, of course faster. They're dying even faster than they would in mild conditions. So the pests, the predators really don't like the heat. And when we look at their predation rate, okay, how many mites do they eat every day? While they maintain a sort of like a normal ratio, if the mild temperature of 22 degrees of 25, when you go into extreme temperature, at first, again, it activates their poikilotemp. It also activates their physiology. They're eating more, but very fast their feeding is dropping. They can't deal with the heat. And to be honest, in this experiment, they lost a lot of them just because they tried to run away, constantly tried to run away from the arena. Get us out of this heat, you know? So yeah, the predators are failing. So again, in Israel, we do what Israeli farmers do. We spray like crazy. But then we got a lot of reports in that the pests are becoming more and more resistant. And I went to see, because you know, I don't know, you deal with growers and they're like, nothing is working, everything is terrible. You have to document it to see if it's really the case. So we went out and we sampled for the last 3 years, samples from all over Israel. And we have here, we tested them here, I'm just showing I think something like 10 different pesticides. And we tested them and we show that we find more and more and more populations that are really, like the farmers are saying, resistant to everything. And we saw, I'm not showing it here because it's not statistically significant, but we do see a trend. So the hotter it gets, the more resistance we have. Which is now, finishing the introduction, now I'm going to talk about these. What we were trying to see, what is it the heat does to the physiology? Do we get more resistance when the pest is exposed to heat? Can it be? Well, even before I start telling you about the test, we were thinking, how are we going to even test this? Like, this is something I did with Professor Shai in Tel Aviv University. How do you even start? Because if we rear them from the beginning in hot conditions, it's already going to affect the development. We are talking about adults, we're talking about pesticide resistance. So we decided, okay, let's just do that. Let's just focus on that. We took adult females, we exposed them to very short extreme heat, 48 hours, that's double the temperature that they were in. But we did take these from actual data that we collected, for instance, in the Gaza Strip, not in the Strip. But yeah, so some days, especially during spring, can come between 25 degrees at night to reach something close to 50 during the day inside the greenhouse of the tomatoes. So we said it's not entirely like detached. What does it do? So now we just tested these 4 populations and just sprayed them with water. And you see if you just spray them in water, we're like this, maybe 5% of them die. And we add in some heat. Let's put both of them. We did heat before and after spray, but why should it matter? It's water. So if we do have this TT, it seems like really sensitive to heat, we get up to 20% mortality. Okay, so already the heat has an effect. What if we add in some spray? So we spray them with a pesticide that activates mitochondria. Let's not go there because there's no time. Again, this is the mortality without the heat. We have high sensitivity relative actually to this one. Once, even without the heat, we got up to 80% mortality. I said we chose one of the more effective pesticides.
ענדי 3 דקות.
3? Then forget. just wait. I just go to the result. Okay, never mind. But I'll go for it. So to show you that, yeah, what we saw is a very nice effect, but here it's not really significant. The timing of the heat stress is affecting the resistance. Okay, you don't see it very clearly here. Let's see it, and we decide to focus on this ATV population, which is the most resistant, and then we will see it the nicest. Okay, when we give them the heat shock after we've sprayed, we really increase mortality. And to be honest, growers got to this a bit naturally themselves. So what growers have started to do from observations in the field is spray either late in the evening or very early in the morning, and then you get in the noon and the afternoon heat shock, and you see an increased mortality. And this is exactly what we're seeing in our experiment. The heat does affect the resistance ability, but in this case we found a sensitivity actually. And because I was talking too much and I have no time, I will show you how it can actually also improve resistance. So we're going to skip all of that and pretend we didn't see them. And just go to the summary and say that, yeah, we still suffer from a lot of outbreaks of this two-spotted spider mite in Israel over summer. It's probably mainly because the predators are not doing what they should be doing, but there are some mitigating factors that we can, for instance, the timing of our spraying. We can play with this and maybe get our pesticide to work better, but not always, as I haven't been able to show you, not in all cases, not in all seasons. And I think in that, I'll just say thank you.
Yeah, so thank you for staying on time. But it was very. But now we have time for a question. So.
No, I think I talked too fast.
Does it have something to do with the effect of spraying? I mean, the efficiency of the chemical in different hours? So maybe the chemical is not efficient
if you
in
ask
certain
so a lot of
temperatures?
the chemical companies, they say that it can last up to a week, even at 35 degrees. But for sure, it doesn't. And for sure, there are a lot of less pesticides that you can spray in the heat because of some of them they are phytotoxic in the heat. So you have a smaller range.
אפשר שאלה בעברית?
אפשר שאלה בעברית?
Yeah, I will translate for the audience. So yeah, sure.
אז איך אפשר להתמודד בשטחים ענקיים עם, כמו שכתבת, אויבים לחקלאות?
So I would just say that the question was how can we actually take that and translate it to fighting natural or enemies or pests in agriculture. Yeah.
So what I'm showing you actually is in greenhouses. In greenhouses, natural enemies are far more effective than they are in the open field. In open field and orchards, we have a big issue because the natural enemies, they are so fast, they go wherever they want. And to be honest, that's an entirely different problem that we're facing. In orchard, we're far more likely to spray pesticides than to use natural enemies. Because even if you spread them, they're going to go away. And here we have like the net or something that keeps them contained to a certain degree. But yes, that's a lot. Main causes.
Okay. You basically say that the change in temperature or the heat wave or the heat stress is affecting the pesticide effectiveness.
Yes.
But did you check the plant's response to the higher temperature? Because like we always say, there is stress, and when you have stress, you fight problems less efficiently. But here, the plant is causing stress on the predator, on the mite. So maybe this is why the pesticide is working better.
That the survey we've done was from something like 10 different plant hosts. So for sure, this is happening everywhere. The survey happened in all vegetable crops and flower crops. So basically in everything that you will grow in a greenhouse in Israel. It didn't go to open field and it didn't go to trees and things like that. So if you already are facing different plants, I'm thinking that the plant effect is probably not the issue here. Because although we are seeing, now that we've accumulated so much data over 3 years, we can see that our trends are different, but it's more that we see differences between vegetables and flowers. And again, I don't think it's because of the plants itself, it's because of the spraying regime. In flowers, it's not an edible thing, they spray like crazy. You don't want to know. So I think it's that. But you're right that we didn't want to get into it. That's why we also did the stress only on the mites and only a very short one. If I had to rear them from the beginning in high temperature conditions, and of course also the plant itself is different, and then you get a whole cycle here. We really wanted to isolate and look only at the mites. and see the response. So yeah, for sure, there's also other studies that we're doing, and for sure, for instance, with tomato cultivars, they really react very differently in different temperatures.
So I'm sorry that I'm stopping it now, but we need to continue. But Adi's here, and I'm sure that she will be happy to answer you later on. Just catch her after we finish, okay?
Thank you.
So thank you very much again. And we are going to move back. I think that now we have Norah Saabna, right? From Haifa University. And we said that the best way to do it is from here. Yeah, that's okay. So, hello everyone. Today I want to show you my study, which is about the summer flowering desert supports local parasitoids, prospects for conservation's biological control. Yes? Okay. So, first, why desert? Desert inhabits over 38% of the global population, mostly in developing countries, and it covers around 41% of the world's land. Deserts are expanding also due to human overexploitation and climate change. So what's unique about desert agriculture? First, there's a limited water and it requires irrigations, and there's a salty soil. So there's two different agriculture in desert. The first one is the traditional oasis agroecosystems that they rely on irrigations from local springs. And the second one is the modern dryland farming systems that rely on water desalinations and drip irrigations and treatment for saline soils. So we're gonna do a biological control in desert. So in a simple words, the biological control means that we use the natural enemies, which could be predators, parasitoids, or pathogens to manage pest population. And in this study, we are focusing on parasitoids wasp. This is the life cycle of parasitoids, as Morin mentioned before. The adults wasp eat sugar from the nectar from the flowers, and they, after mating, they lay their eggs inside the host. They develop inside the host or outside, and then they emerge to be adults and kill the hosts. So the hosts represent the pest, and the parasitoid represent the natural enemy. So there's three different kinds of biological control. The first one is the augmentative biological control, which is rearing the facilities produce natural enemies, which are then releasing into crops to support pest populations. And the second one is the classical biological control, which is introduce natural enemies from pest native area to control it where it's become invasive. So the last one, which is we are focusing on in our study, which is conservation biological control, which is the pest management approach that focus on enhancing and effectiveness the abundance of the natural enemies that already present in the environment. So there is a challenge for biological control, especially in dry lands. For each one of the biological control, for the challenge of the augmentative biological control, that it requires rearing facilities and needs distribution system, which is also super hard in deserts and dry lands, and it requires trainer personnel to monitor it during the seasons. So the challenge of classical biological control also requires importing and takes a long time. So the conservation's biological control, also there is a special challenge in deserts, which is a water shortage, limited planting, supporting vegetations. So the lack of the water, because of the lack of the water, prevents farmers from planting extra non-vegetation crop in order to support the natural enemy that already exists in the field. So
we suggest solutions to planting a desert tree that adapt to deserts, such as a Sisyphus, could offer a particular solution. So Sisyphus, and why Sisyphus? Sisyphus, which is Sisyphus spina Christi, which called in Hebrew Shizaf Matsui, and in Arabic is Shajar al-Sidr. It's well studied for afforestrations in desert due to the adaptations. So there's a low water requirement and heat tolerance, and there's economic significance. This tree produces a very high quality honey, and there is also very significant ecological significance. They produce fleshy fruits consumed by birds and grazers. And the most important thing that they produce nectar. That's a very important source during summer, because the peak of the flowering in this tree is in summer. So this leads us to our study questions. We have 3 questions. The first one: what characterizes the parasitoids community on Sisyphus spina Christi? And do these parasitoids feed on trees' nectar? And then, are they potential agents for biological control on nearby crops? So in order to answer these questions, we sectioned samples from different sites, as you can see in the map, from Khuwailid, Nakhal Tavor, Rishafim, and Tirat Tzbi. Each one of the sites that we choose have a different average annual rainfall, as you can see in different color. We sectioned around 15 trees from each site, and we identified the parasitoids in lab. So here are some of our results. We can see in this graph the total parasitoid abundance along the rainfall gradients. We can see that there's no correlations between the rainfall gradients and the parasitoids abundance, and also there's no correlations with the flower abundance, as you can see it in the orange line. And there's no correlations also between the flowers abundance and the rainfall gradients. So we found something, a lot of parasitoids that lives in these trees in different sites. We identify 3 dominant species. The first one is the Gramosoma ramutensis, which is represent over 50% of the whole parasitoids that we collect. And we can see the peak of the Gramosoma in Nakhal Tavor. This is a leaf miner Lepidoptera. The host is a leaf miner. And the second dominant parasitoid is Glyptabantelis, as we can see it in the graph in orange. And the third one, also the host of the Glyptabantelis, is the leaf miner. The third one is Gnabutodon. We can see it in yellow in 2 sites, in Tirat Tzvi and Rishafim. It's also the host is also leaf mining Lepidoptera. So we this graph shows the parasitoid abundance across seasons in specific sites, which is Nakhal Tavor, and we can see that the peak of the parasitoid abundance is in July, in summer. Also, it's nicely coordinated with the peak of the flowering abundance, as you can see in the orange line. So it's not just that. We want to make sure that this parasitoid that we call parasitoid waste that we collect from the trees already eat sugar from the Sisyphus. So we use antron, which is a yellow liquid that changes its color when there's sucrose or fructose. So we just add 1 drop of this yellow liquid antron, and if it's turned to be yellow, it means that this parasitoids already eat from the Sisyphus tree. Here's some results. We can see that in this graph, the sugar feeding frequency across seasons. We can see that the peak of the proportions of antron positive also in July, and it's nicely coordinated with the flower abundance, as you can see in the orange line. So here's some answers. What parasitoids wasps live in Sisyphus spina Christi? So the dominant parasitoids attract leaf miners parasitoids and are the most abundant in summer, since summer is the most challenging season in desert, and it's with just a few flowers blooming around. And do they feed on trees' nectar? So probably yes. And are they potential agents for biological control and nearby crops? This is one of our future plan, that we will target leaf miner pests of summer crops. So here's some of our future plan, future work, that pest control in the pest control experiment. We will set up organic tomato plots next to potted Sisyphus tree, as you can see. And then the tomatoes, why we choose especially tomatoes? Tomatoes are highly valuable summer crops that attract many arthropods, pets, including leaf miner. And then we aim to test whether the parasitoid wasp move from the Sisyphus into the tomato field and improve the fruit's quality. This is our future work. Thank you for listening.
Yeah, so we know that we need to come back to this conference in 2 years to hear about the next. Any question about what we heard? Yeah.
So, because you are looking only on one point of view, that is the parasitoids. So if you look on the other herbivores, so it might provide a refuge to the other herbivores which will be living during this time. So,
Okay,
to solve one problem, you might create another problem.
Yeah, so maybe you mean that this tree maybe also support the herbivore also, the pest in this situation. So maybe it could be. It's a general problem in general to augmentative in conservation biological control. It's not a specific question. It's general for this kind of biological control. So the answer is yes, maybe. Maybe we need to check that also. Yeah.
More questions? So if not, then thank you again. To our final talk of the day, which will be by Morin Mishra from Ben-Gurion University.
Hello everyone, my name is Morin Mishra, and today I'm going to present part of my master project. And the title of my presentation is Driving Defense Responses in Certiaria plant in response to aphid infestation on its different developmental stages. So in the natural ecosystem, plant, animal and insect exist together, and they show different kind of interactions, like positive and negative interactions. In the case of negative interaction, usually with insect, it's feed on the plant and it causes massive agricultural yield losses. But in the course of evolution, plants also evolve different kind of defense responses or mitigation system to overcome these challenges. And for our understanding, we divide it into two type of defense responses, for physical and chemical defense. In the case of physical defense, it's produced barrier with the help of trichomes, cuticle formation and callus, whereas in the case of chemical defense, it's produced toxin chemical which act as a deterrent to this kind of insect and prevent infestation. Though the Though there's lots of study done on this kind of defense system, but they are mostly focused on certain defense system or certain developmental stage of the plant. So there's need to study, there's need for the study like how these defense responses are varying over developmental stages, whether it's constant or varying. So to address this research gap, we used Ataria aviaris plant. It's C4 plant, belongs to family Gramineaceae, and because it's have a short life cycle, it's completed its life cycle in 8 weeks. So it's make it as the best candidate for our experiment. And for infestation, we use our Arpadi aphid, which are a modern insect and known to feed on the Gramineae family. So plant is sessile organism. It cannot move one place to another. That's why it's a limited resource for their growth and defense. It's need to trade off between that. So when whenever plant undergo different developmental stages, it's reallocate more resource towards the growth rather than defense. So that's why we hypothesize that the chemical and the physical defense will vary over different developmental stages of the plant. And to prove and disprove this our hypothesis, we asked 3 questions. That is, how these defense responses are varying, what is gene expression pattern alteration, and then last one is like whether how the aphids are performing on the plant. So and keeping this all in mind, we designed this experiment where we infest plant every week with respect to different developmental stages with aphid for the short and the long period of infestation. And today I will only talk about short infestation versus control. In short infestation, we use clip cage and 4 days of infestation, and then we use it for further analysis. And this is how the experiment looked like. So we started with one simple experiment: how aphids are performing on the plant. What's the aphid population growth rate? And what we found in early week, we got less aphid population growth compared to later stage. So then we asked question why this is happening. Then we checked we checked plant defense, physical defense responses, and we got in early stages of development, that's 3, 4, and 5th, we got significant high induction of trichome, whereas in the later stages we do not get any significant induction. And when so I read in literature, but I found when leaf get expand, so epidermal cell lose ability to divide. So that's why we are not getting induction of trichomes in the case of physical defense. And there's also different other physical defense also present, so maybe it's helping to mitigate the defense responses. And then we check gene expression pattern. And as you can see here, in early week, that is week 4, we have a higher upregulated gene, and then it's gradually getting reduced in later developmental stages. Okay, so now then we try to see whether these genes are doing which biological activity. So we found phenyl coconut biosynthesis pathway are activated throughout developmental stages in response to aphid infestation, and this pathway are known to production of lots of secondary metabolites or chemical toxin which act as a deterrent for aphid or any kind of insect. And when we see the gene expression, you can see though it's activated throughout developmental stages, but it's have a higher expression in early stages, and you can see with this graph as well the mean fold change. And then we also got different kind of defense responses as well in early stages. That is hydrogen peroxide catabolic process and jasmonic acid biosynthesis and signaling pathway. This is one of the important plant phytohormones which plant produce in response to any kind of biotic stresses. Here as well you can see in early stages there is activation of biosynthesis and transaction pathway of jasmonic acid. And for our understanding and for to understand the pattern or to remind it, I again plot it with the mean graph. You can see in early week we have a higher expression of jasmonic acid biosynthesis transaction pathway compared to later stage, that is week 7. then we also checked serotonin, which are known to, which are one of the important secondary metabolites or part of chemical defects in Satire everidis plant. And this pathway are already, also characterized by one of my lab and she also checked the exogenous application of this, and it showed that it's negative effect aphid survival rate. So that's why we checked it, and we found it's activated throughout developmental stages of the plant, irrespective of it, when we are infesting. And that's lead to accumulation of the serotonin. So this this helps us to understand that why we are getting low aphid population growth in early stages compared to later stages, because the plant in early stages are more defensive than the later stages. Now the second part where I will talk about down-regulated genes, in which pathway it's involved, like what it's doing actually. So here we found all the down-regulated genes are mostly involved in photosynthesis and carbon fixation related pathway. And you can here, as you can here see all the component of the photosynthesis activity and are getting down-regulated in response to aphid infestation in each week, and which also lead to degradation of chlorophyll. Let's say here, there's significance here, but in later stages we don't get significance, but you can see a pattern is getting reduced compared to control. And here you can also see compared to control one, you can see it's getting yellowish. But though there's low photosynthesis or there's leaf yellow, but we got high amount of sugar, carbohydrate, and amino acids. That's totally opposite. Then we did literature survey and we come across this research paper where they mentioned that this premature senescence in the leaf, it's act as a defense, one kind of defense mechanism. So we checked in our data as well, we found some of the senescence inducing genes are getting upregulated. It's higher in big 3 because in control we don't supposed to get senescence in early leaf, but in later we have like then senescence are started, that's why we don't get significance, but there is senescence. And you can see also a similarity in this plant and their paper and our paper, we also get yellowish. So what actually happening here? So whenever plants get infested with aphid, it's at the site of infestation, it's upregulated senescence inducing genes, and then senescence gets started, premature senescence gets started, and what it's do, it's degrade or just break down all macromolecules. That's why we got at the local site of infestation high amount of sugar and amino acid because of breakdown of big sugar and the protein. And then it's redirected to other part of the plant for development of new defense compound. So this is all I have. So early stage of the plant show higher defense responses, as we saw in physical and chemical defense are more activated early stages, whereas the chemical defense are activated throughout, but it's have a more severe effect on early stages compared to later stages. Yeah, that's that. Thank you. Thanks my professor Zin and all my lab mates and my all friends, like they are always there for me and help me for writing my thesis.
So thank you, Anmol. Questions? Yeah, Mite.
Hi, thank you so much for the talk. I kind of didn't understand the role of serotonin. You mentioned that serotonin is important for development of the plant, but I didn't really understand what's going on there. Could you really explain it again?
So in humans, serotonin is like happy hormone.
Yeah.
But in plant, it's defensive compound. Like so, when we start with this our project, we try to see like which secondary metabolites are involved in serotonin, like in our plant as a defensive compound. So we checked multiple like gramine, benzoxenoid, and then we found that serotonin is the compound which is synthesized in response to aphid infestation. And then she checked like exogenous application of serotonin. So it's have negative effect on aphid survival and aphid performance. So that is why in our data again we checked because the Sataria, and we check how it's behaving, whether it's also activating higher in early stages compared to later stages, but it's activated throughout. Like when each week when we are infesting with aphids, we are getting high serotonin.
So essentially kind of like how testosterone has a double function in the human body, where it's both like a male hormone but also an important defense hormone, and also the female hormone, I forgot what it is right now. Then you're saying that the serotonin has both the function that we classically associate in the human body, but also an important defense one.
Yeah, in plant it's defense, but
Yeah,
it's also there's also like further it's also converted into melatonin. I'm not exactly it's something.
But it's a cascade from the jasmonic acid, basically.
What?
A cascade from.
No, it's not. It's totally different pathway.
Okay.
Just regulate. So does it have any influence on the plant itself? Serotonin, so it's synthesized totally as a secondary metabolite with no influence on the plant itself.
It's have also influence on the plant. So it's helping plant to go under reproductive stages, and also it's also convert into another like downstream pathway, and it's have like helping plant development. I don't recollect right now the whole actually the role, but it's have also role in development. It's like
so we should infest plants with acid and have more serotonin in our body.
You can use
find a solution for
The question is if
instead
it's
of
bioavailable.
no?
The question is if it's bioavailable.
it's available, but
Other questions? Okay, so if not, Anmol is of course here to continue, and I want to thank all the participants of this session and the former session, and everybody who came to listen, and lunch. So.